Hi, everybody. Welcome. We're really happy to be here because this is a very important topic and a problem that is rising in this area, as you will soon find out. So that's my book, by the way, which is for sale in the alcove back behind us. First, a little bit about what this epidemic is not, because there's a lot of misconceptions about it. It may not be what you think, what your initial kind of impressions have been, that it's easy to diagnose, easy to treat. It's not necessarily what doctors have been told as to how they pick up Lyme disease, as to how they resolve the problems of Lyme disease. It is not small, and it's not under control, frankly. But first, the problem. Let's define it. Lyme disease is soaring in the United States. This chart here shows about 150% increase since about 2000. Then we'll zero in to New York State. And you see Lyme disease in that same period rising about 80%. It's spreading more widely around the United States, so that's why it's the US numbers as a whole are rising more. But now let's look at your neck of the woods. Where you live, Lyme disease is soaring. Central New York has seen a 300% hike in Lyme disease numbers just since 2008. Those other numbers you saw were back from the early 2000s. So just since 2008, it has come on strong. About twice as many cases in that period in Onondaga County, where we are now. About a 20-fold increase in Oswego, just to the north. This is an interesting map that ran with an article that I wrote in 2012. And I just put it up there to show you that big red blob there. That's the Mid-Hudson Valley. At that time, the Mid-Hudson Valley, where I was reporting from, had the highest rates in the state and in the nation, and I've since learned pretty much in the world. If I was to do that map today, and by the way, the darker the color, the higher the rates of Lyme disease. Back then, you guys were sitting pretty. Very, very low rates of Lyme disease. Now you'd be more in the dark blue category. And why is this? The infected ticks are here. Roughly about half of adult ticks in each of your local counties, ranging from maybe 40% to high 50s, are infected with the Lyme disease pathogen. And beyond that, I have my little ticks. Some ticks are also infected with other things. There are other pathogens, other bugs, and other illnesses that one tick bite can deliver. The leading secondary infection that they are finding locally is anaplasmosis. It's another bacterial disease, can be very serious, especially in older people. But they're also finding something called Borrelia miyamotoi. We don't have a test for that. Doctors don't know to look for it. This is a little bit of the story of Lyme disease. We're behind the 8 ball. We haven't caught up yet. Quickly, I call Lyme disease the first epidemic of climate change. And climate is a key driver here. We know that things have changed in New York State. It's 4.4 degrees warmer in the winter here than just in 1970. We're seeing a lot of changes. Trees bloom on average 10 days sooner. Bees pollinate earlier. We're seeing fish stocks move up the Atlantic coast. It's warmer, and that's a good thing for ticks. And why do I have this here? These are migratory flyways for birds. Birds have long carried ticks hither and yon, north and south. We've got these flyways from Argentina in the south to the Yukon in the north. They've often, carried the ticks, dropped the ticks. But now when the ticks arrive, they find a very, very hospitable place to live. This is a mouse. This mouse looks a lot different than a mouse might have looked 20 or 30 years ago. You see all those ticks on its ear? The mouse is the first place a baby tick goes to get its sip of Lyme disease infection. Then, when it goes to its next life stage, it's looking for another mammal, and sometimes that other mammal is us. So basically what we have is this changed world. We have lots of cut-up forests that really support mice, that don't have much biodiversity, so you don't have foxes to gobble up those mice and keep things under control. We have a lot of deer, we have a warmer climate, and we have a perfect storm for an epidemic. And by the way, we did this. We adulterated the planet. We made this possible. And moreover, we haven't met the challenge. And this is where I'll get into some of the health and diagnostic quandaries. Just a little bit about myself. I'm an investigative reporter. I came into Lyme disease blank slate. I had some impressions, but I wanted to find out what the story was. I didn't have chronic Lyme disease. I was an objective reporter. And it took me a long time to get to Lyme disease because as an investigative reporter, I look for these kind of things. Corruption and waste of money and law breaking. I didn't expect to find that in Lyme disease. I found an awful lot of it though. So this is one of my first stories. That's the headline. It's about antibiotics, but it might as well have been that. Huh? Okay, so here I am investigating Lyme disease. I find that we have the highest Lyme rates, possibly in the world. But what are the powers that be concerned with? What are officials talking about? Not about controlling the ticks, not about patients who had to go to 5 and 10 doctors to get diagnosed. No, their focus is on antibiotics and on doctors. Doctors who are trying to figure this out. Almost every doctor who treats Lyme disease as a specialty in New York has been investigated by the Office of Professional Medical Conduct because they're breaking the rules. Okay, how we got here and why those doctors are trying to figure this out. There are myths surrounding Lyme disease that are very dogmatically upheld. The first one is that the test works. The second, 2 to 4 weeks of antibiotics and you are cured. You don't have that Lyme disease spirochete in your body anymore. There's a lot to challenge that. This is just a chart on Lyme testing. Holly will talk a little bit more about that. But this shows how well it works early on, maybe 40% of the time. As you continue on in the infection, as the spirochete disseminates, moves throughout your body, the test gets a little bit better. Still not perfect. And by the way, you're allowing a very dangerous bug to advance throughout your system. This whole paradigm of just wait is flawed and harmful to patients. And by the way, when doctors look at this, they see that last one, 100% late disseminated, we're fine. So we've also got a problem with the treatments, which primarily are early on doxycycline. 10 to 20% of patients stay sick for some period of time, weeks, perhaps months after treatment. That amounts to, based on the 2017 numbers, 42,000 to 84,000 people in 2017. And there's a big caveat that I have to mention here when I say 10 to 20%. That's among the people who are early treated. What about the people whose diagnosis is delayed, whose infection progresses? Yeah. So this is how we got where we are now. We have not done the work on Lyme disease. This is the number of clinical trials that we've done on a variety of different diseases. Up at the top for HIV/AIDS, which, by the way, the pathogen for AIDS and the pathogen for Lyme disease were identified in the early 80s, about a year apart. About 600-700 clinical trials. We've had 4 for Lyme disease. And the reason we've only had 4 is because the mainstream medical literature maintains that we have solved the problems of Lyme disease. We know how to diagnose it. We know how to treat it. There's been no urgency to attack Lyme disease on a large scale. And this, again, is the upshot. This is how much money we're spending for Lyme disease. It's wrong. So this is me 30 years ago, standing in a field, knee-high in grass. Do you think I would do that today? I have a new relationship with nature. I look at the field behind my house with a different eye. It's unfortunate, but it's necessary, and you have to do that as well. You have to see the ticks that are lurking there and take precautions against them. This is where I get to show off my grandkids, because we need to protect children. Children from 5 to 9, that big blue bar there on the left, that's boys 5 to 9, are most frequently infected. Children in general, 5 to 14, most frequently infected. Kids lose months and years of childhood and schooling to Lyme disease. So, a few little tips. Be vigilant. Be aware that ticks are out there. Stay in the center of a path. Do not brush up against tall grass or weeds or leaves. Cover yourself. Check yourself after being outside. And be your own best advocate if you think you have Lyme disease. That means demanding treatment, demanding testing. If you're still sick and the test comes back negative, you demand to be treated. Read up on Lyme disease. There's lots of good resources. LymeDisease.org, there's a ton of good information out there. Am I good? Anyway, there is a little bit of good news. This is the World Health Organization's list of diagnostic codes for Lyme disease. It's kind of a before and after. Within the last year, they added 12 additional codes for Lyme disease. They went from 4 to actually 15. One of these were dropped. But anyway, it's a good movement. We're starting to recognize that Lyme disease can do more than has been acknowledged in the past. Lyme dementia is one of the new diagnostic codes. So anyway, I'm going to pass this off to Holly, and I thank you very much.
So thank you, and I actually, before I start my time, I would like to just tell everybody that if you haven't read Mary Beth Pfeiffer's book, you should read it. There's a lot of information in there that you can, is very useful, and if anybody's interested, we have it at our booth, which is the Lyme Action Network booth, which is kind of around the corner, and Mary Beth kindly signed several copies of the book for you too, so if anybody's interested, you can find that over there. So I'm here as an academic scientist. I'm a professor of microbiology. I am a researcher actively trying to identify a diagnostic test that is more accurate than the ones that we currently have available. And as my daughter calls me, I am a provoked mama bear, because my daughter became mysteriously and suddenly ill after, with basically no background whatsoever, no reason to assume that it was Lyme disease. And the journey that we took to get her diagnosed, to get her treated effectively, to get insurance companies to pay for any of it, that really provoked the mama bear in me. And so although my daughter is now doing well after a 2.5 to 3 year journey with Lyme disease, the mama bear hasn't calmed down. So I'm doing what I can to help other people, which is to be an advocate for change. And unfortunately, since it's not coming from the medical side, or not entirely from the medical side, we need legislative help for that too. So some facts about Lyme disease that may or may not ever come through the sort of the major message that we hear from public health organizations. According to CDC statistics, there are over 400,000 new cases of Lyme disease occurring every year. And just to put that in context, that is the second most common infectious disease of all infectious diseases tracked by the CDC. Chlamydia is number 1, Lyme disease is number 2. So this is not a simple tick-borne illness. This is a major infectious disease that is being sort of overlooked because the consequences of long-term disease are being also overlooked. The majority of patients, the majority of those new cases of Lyme disease will go on to develop a debilitating chronic illness. And by majority, I would say the lowest end of that estimate is 60%, and the higher end would be 90%. And I'll explain to you why that is true. But when I'm talking about a chronic illness, these are the symptoms that are associated with those illnesses. Some of them affect the neurological system, some of them affect the joints and muscles, some of them will cause profound fatigue. Many of them are related to sort of malfunctioning of the immune system and the central nervous system. So, by the way, the 427,000 cases is because the CDC readily admits that they cannot accurately count the number of diseases. So when they provide you numbers, that is for tracking disease trends, you can automatically multiply those numbers by 10, and that is something that the CDC endorses. In 2018, I was very honored to serve as a member of the Tick-Borne Disease Working Group, which was convened by HHS, the Department of Health and Human Services. We worked for a year I was on the Testing and Diagnostics Subcommittee, and we worked for a year to examine the issue of Lyme disease, do a deep dive in that study, put together a comprehensive report, and in December of 2018, we delivered the report to Congress. Congress now has a platform upon which to act, and of course, it is up to us to tell Congress we want them to act on our behalf according to the report. You can go read the report. It is public at that website. At that link right there. And one thing that came out of it, maybe you saw the press releases that went out with this too, is that there were 3 major areas where significant gaps were noted. Gaps in research, gaps in understanding, gaps when it came to patient issues, and those are the issues of prevention. We are failing to prevent Lyme disease, obviously, because the numbers keep going up and up and up. In the area of diagnostic testing, where the consensus of the body that I was on was that the current types of diagnostic tests for Lyme disease are not clinically accurate. And what that means is that you are equally likely to have Lyme disease whether your blood test result comes back positive or whether your blood test result comes back negative. You are equally likely. So the clinical utility of a diagnostic test, meaning who to treat and who not to treat, is very, very poor. And that's one of the areas where if we had had an accurate diagnostic test, back when my daughter was bitten by that tick, which were years before she actually broke, we kind of put it that way, none of this would have happened. I wouldn't be standing here talking with you now, and many of you would not be in the audience right now because I presume many of you are here because you have had similar experiences. And number 3, there are significant gaps in the areas of treatment. And we know from a growing body of research on the microbiology of the tick-borne microbes that they are antibiotic tolerant, that a single short course of antibiotics is effective for a marginal number of people, but not for the vast majority of people. And so we need to do research looking at different combinations of antibiotics, different treatments, different treatment approaches, and that is desperately needed. So these three areas were, of course, the major focus, but I want to say I focused in on the diagnostic test because the diagnostic test, I think, is, until we have a way to say who does have this disease and who doesn't, we really can't design clinical trials, we can't do research, we have very few options with that. So trying to pare down the problem, it's a huge problem. There are so many areas, but if you pare it down, only a minority of Lyme disease patients can be accurately diagnosed. The only group of patients that can get an accurate diagnostic are those that develop a bullseye rash following a tick bite. I call that group the lucky winners of the bullseye rash. Lucky you, because if you have a bullseye that is a perfect bullseye, then the doctor will say, oh, you have Lyme disease at the moment of your tick bite, and treat you with an antibiotic that will stop you from, or marginally stop you from going on to develop secondary chronic symptoms. That is the only group. That is 10%, roughly, of the population, of the entire population of Lyme disease. A recognizable rash occurs in 10% of patients. I'm saying that as on the basis of this article right here. I'm saying that on the basis of studies that were done with non-human primates, where the rate of the perfect bullseye rash is 10% or less. It's, for any rash, it's 50%. So what the skin rash is a skin infection. And these bacteria go way beyond skin. Once they're in the bloodstream, they disseminate or spread to tissues in many other parts of your body. And so only those people with the bullseye rash receive early antibiotic treatment. Only those early antibiotic treated patients are the ones who have the highest chance of not developing secondary illness. So this is right here the diagnostic algorithm that is used in medical practice to determine if a person has a case of Lyme disease. Does anybody feel like they need a PhD in something to be able to understand this algorithm? Anybody else besides me? Okay. So I want to show you how this algorithm fails to diagnose Lyme disease, beginning with the early criteria. It says Lyme disease is suspected. So where is it suspected? It's suspected if you live here. If you live in the Northeast, if you live in a region in the upper Midwest, or if you live in a few places in California, physicians are aware that patients have Lyme disease, and if you don't, then you're not going to meet that first criteria. So that excludes 80% of the country from accurate diagnosis. So then let's say you live in the right place. Okay, so how many people, once again, get the bullseye rash? So the next criteria the doctor looks for, is there a bullseye rash? Okay. Less than 10% of Lyme disease patients get the bullseye rash, but if you do, you get a diagnosis of Lyme disease. Your treatment is 3 weeks of an oral antibiotic, and you are presumed to be cured. But what Mary Beth alluded to was that 10 to 20% of that subgroup of early diagnosed, early treated patients still go on to develop chronic disease. So obviously, that is not a fully effective treatment. 20% of 400,000 people is 80,000 people a year developing chronic illness. Wait, I think I did the math wrong, but you get the picture, right? That was right? Okay, good. So if the bullseye rash isn't present, then what does a doctor do? So the next step is to perform a test called an ELISA. What you need to understand is that ELISA test is only half accurate. It's 50% what is called sensitive, meaning that here's the test where you are equally likely to have Lyme disease, whether that test is positive or negative. Antibodies are not produced by your body in response to that spirochete for up to 6 weeks post-infection. So if you have a tick bite, a week later you go to the doctor says, well, there's no rash, let's do the blood test. Blood test is negative. You don't have Lyme disease, when in fact you could possibly have Lyme disease, but the test is not accurate at that early stage of the disease. All right, so your test, let's say your test turns out positive by some miracle. But let's look at what happens if it is not. Okay, we'll look at that pathway first. So if your test is negative, what does the physician do if your blood test after a week comes back negative? And the answer is wait for 6 to 12 weeks to see what else might happen, to see if the bullseye rash occurs, to see if you develop flu-like symptoms, to see if something else occurs. So while that antibiotic treatment is delayed, here's what the bacteria are doing. They are in your blood, They are swimming as fast as they can out of the blood into tissues. That would be your heart, that would be your brain and central nervous system, that would be your joints, where they establish permanent colonies. They live there, they feel their job is to live with you forever. They establish permanent colonies, and in this form, the bacteria are antibiotic tolerant. That's more than resistant. It means they could care less whether you treat them with antibiotics or not. At that stage, treatment becomes a lot trickier because it's not a short course of antibiotics is not going to eradicate them, and even it's questionable whether IV antibiotics or longer term antibiotics are going to be able to either. So after 6 to 12 weeks, let's do the test again. Let's say it comes back positive, or if the ELISA was positive, then you need a second test. That test is called a Western blot, okay? Oh, and by the way, if it did come back positive, you would have gotten, again, 3 weeks of oral antibiotic treatment, even though you could have been infected for months to years, okay? So this is where things get complicated, because at this point, if the test is negative, then what happens if the test is negative? And the answer is you get diagnosed with something else, you get diagnosed with nothing, you get no treatment. This is what the disease known as chronic Lyme disease is. This is not early diagnosed, early treated, still treatment failures. This is treatment failure because you've had the infection for a year or 2 years or 5 years, where the bacteria are now permanent and much more difficult to eradicate. It is also, in the case of many physicians, still follow the rules that there's 2 classes of antibodies, one is early, one is late. And if you are positive for this test, using one type of antibody, one class of antibodies, then the doctor may say, well, your tick bite was 3 years ago, this is not an active case, this must be a false positive. So positive blood tests can still be interpreted as negative because physicians are not aware of how remarkable these bacteria actually are. And what I want to point out is that treatment failure, whether it's early treated or late treated, leads to chronic disease symptoms. This is a paper that was published by Johns Hopkins University in which the population of patients that had these chronic symptoms following early treated Lyme disease were characterized. They looked at their blood, they looked at their bodies, they looked at their symptoms, and they created a paper, a profile, if you will, of what this entity called post-treatment Lyme disease syndrome is. This is 10 to 20% of the 10% of people who developed the bullseye rash still are treatment failures. So notice that it is a real, what the authors of this study say, this is a real disorder in the absence of a clinically detectable infection. What does that mean? It means the blood tests come back negative even though you still are infected. So post-treatment Lyme disease, I'm calling that subgroup 2 for the moment. And I also want to point out that treatment failure following long-term illness, meaning in other words you were infected 2 years ago, now the treatment starts, you will end up with chronic symptoms if that is the case. That's what happens because the bacteria have been established. 3 weeks of oral doxycycline is not going to effectively treat an infection that has been in your body for 3 years. And so as a result of that, this article is published by Dr. Richard Horowitz and his team in which he defines that body. He does the same thing that John Alcott at Johns Hopkins did with his population of post-treatment Lyme disease syndrome. Dr. Horowitz calls this MSIDS, for multiple systemic infectious disease syndrome, because what he's finding is that many of the patients that are difficult to treat, it's because they have secondary infections. So Borrelia is bad enough, but then you add Bartonella to that, you add Babesia to that, which, by the way, the standard antibiotics do not kill, and you end up with a very severely compromised immune system, and actually life becomes much more difficult. And notice from the paper, another research paper shows that 42% of this population end up having to stop work as a result of their disability. And so I just want you to realize that that was kind of bad news. I mean, it's sad, there's good news because research is moving forward, and it's moving forward because of the awareness that this is a growing problem in this country. And I have to say, most of the research that I presented to you is being funded by private philanthropic organizations like the Cohen Foundation, like the Global Lyme Alliance, like Bay Area Lyme Disease Foundation, like Focus on Lyme. And with that funding, progress has been made significantly. It's time for the NIH to stand up and start doing their share too, because the data is accumulating to show what's going on. And the answer is, disseminated permanently established infections drive chronic inflammation within those tissues, which also affects your immune system's ability to operate correctly. And so these 3 papers published within the last year show that. Okay, paper number 1 was a study done with non-human primates treated with antibiotics. So with non-human primates, you can infect them and allow the process to occur and look at it weekly, monthly, for a year. And then that is something you can't actually do in humans. Non-human primates are an excellent model for this disease in humans. And please notice that after treatment, monkeys who had been infected and gone through this process with that one single bacteria, Borrelia burgdorferi, the cause of Lyme disease, still had spirochetes present in many tissue sites, both the untreated animals and the treated animals. Then in this paper, this is your brain on fire. And this is the part that in this particular paper, what they're describing is PET scans, so these are scans of the brain. Where you see red means inflammation. And so you can actually see, I'm thinking that's a pointer, on this side, control brains, and on this side, these are brains of people with post-treatment Lyme disease syndrome. Brain on fire. People with that sort of infection with Borrelia burgdorferi have long-standing symptoms because they have inflammation in their brain. Inflammation in the brain leads to a whole lot of downstream issues. And the last paper actually shows, let's do that, that this disease is also associated with another illness called small fiber neuropathy. And the small fiber neuropathy was found in 100% of the post-treatment Lyme disease syndrome patients that they looked at. This is true of the chronic Lyme disease patients as well. And so, just a shout out to my organization as a provoked mama bear, this is what we do. We start advocacy organizations and start yelling at people. That would sort of include you. I'm not yelling at you, I'm just yelling in general, okay? But this is our publication, Learn the Facts, which I guarantee you is evidence-based. Everything that is in this publication can be backed up by the scientific literature. It is available over at our booth, which is around the corner here somewhere. Please pick it up. Please feel to hand it out to all your physicians and anybody else you might possibly know. And I also want to point out that we have an entomologist on our team. This is a bug lady. We call her the bug lady. I'm the micro bug lady. She's the bug person, who has done research on different types of oils that are actually repellents for ticks, right? Because the best way to protect yourself from Lyme disease is to prevent tick bites. And so we sell a product that has the essential oils that she was able to identify from her experiments on torturing ticks, that's what she does. I torture microorganisms, she tortures ticks. And so that we also have that over in our booth as well if you want to figure out how to get there after our talk is over with. So thank you very much for listening, and hopefully we'll have questions at the end.
Green advances it, red turns it back, and there's your,
That's the advance right there.
there's your advance.
Well, thanks for coming out today. I'd like to know how many people have... What?
They put mine third. You go. I'll go first. So I'll
Okay.
make you go. Don't worry, I'll be quick. Okay? Can I have that one? Hey. Thank you very much. Okay, I'm Bob Jagir from Hygienics Labs, and you've heard about how bad lab testing is, and I'm going to show you why it is. So we're going to talk about staying ahead of the plague, because it's getting worse, and I can't even talk to you about some of the tests that are now available, because New York State doesn't allow me to do that. So let's talk about what we have got and where we're standing. Here's 2 pages that anyone can see from the CDC website. The one on the right, Paul Mead gave to me when I was out at the Rocky Mountain Lab about 6 months ago. He presented it about a year ago at Mass General. And the one on the right is going to show you where Lyme disease is going to get worse, according to the CDC, over the next 2 to 3 years. Okay, here's New York State, according to your Department of Public Health, and how bad it is. Yes, anything there that has a percentage above 20% is considered an epidemic in a state by the CDC. So if you're above 20% to 25%, you can expect that you have a full epidemic of your area. Remember, this is all based on data that's about 10% at a 0 next to all those. So now here's the bigger problem. Right now, we know there are at least 7 different strains of Borrelia of the 52 in the United States that we have found that can cause what we call Lyme disease, the sensu lato strains. You hear of one strain, Willie Burgdorfer, the Burgdorferi strain, which is called the sensu strictus strain, but there are others. We have Willie's strain, we have Lone Starrii, which used to be called Master's disease from the Midwest. Yes, it is here. We also have both the Afsidi and Guarini strains. They came from Europe. Thank you, Europeans, for sending us those. And we now, of course, have got a couple other things. We, of course, have the Mayoni strain, which is that's named after the Mayo. Bless their little heart. They found it 3 years ago, so they named it after themselves. Then we have 2 other strains that are going to be called tick-borne relapsing fever that I'm not allowed to talk about because we can't test for them in your state, but be very well advised that if you look them up, there are guys at Columbia and other places that are looking at these. Miyamotoi is almost epidemic here. We do know that Hermsii is an epidemic west of the Mississippi, so please don't go camping out there. And there's going to be more things coming, other strains in the near future. So, Lyme disease progression. You heard a little bit about this. The tick bites. It changes some outer surface proteins on it because of temperature difference between the host and the bug. That increases the presence of Borrelia burgdorferi and multiplies it out from about 500 copies in the midgut to about 200,000 over the course of 24 to 36 hours. And when that bug attaches and you get that little bubble of blood, the first thing in your body that sees it are your what are called dendritic cells right underneath your skin. They pick it up, they digest it, they send out a T cell and an NK cell response to attack it immediately. You cannot test for those in your state. In other states, yes, I can test for them, and I do now have a T cell response test using recombinant proteins that actually works. But you're in New York, you cannot use that test, so you're kind of at a loss. You have to wait to, as Holly said, wait for an immune response to take place, which can take 4 to 6 weeks. So later in the disease, the chronic phases, and yes, it isn't post-treatment Lyme disease, it's chronic late-stage Lyme. What happens is the bug comes out of tissue, out of its cyst form that it's been hiding, changes its surface presentation, your body sees a new bug, but it also picks it up in your macrophages, take it back to your lymph nodes, develop a new immune response, and voila, that's what's causing your IgG and IgMs, more importantly, late in the disease. It is not post-treatment Lyme. It is an active infection that has just found its way back out of your tissue. So, there are a lot of different tests that we over the years have failed at developing for Lyme disease. Here are the ones that are most known. Of course, the humoral or antibody response. Those are the responses your body gives to the bug. They develop antibodies too. And we've got a bunch of them on there, and I'll go through some of the techniques. And then, of course, there's PCR technology and some fish technologies that are now available. But again, you can see in big red things that are not available here in your state. I was at a meeting in Dearborn in 1994. A bunch of us from University of Connecticut were invited. Well, 4 of us anyway. And the CDC got up front and told us what we were going to do to diagnose Lyme in a laboratory setting. When they did that, they told us that we had been doing a good job with hepatitis and HIV, so they wanted a 2-tier approach. 2-tier approach, we stood up at UConn, the guys at Yale stood up at their table. We couldn't sit together, that's a whole different story. But we all stood up and started waving our hands, and that's where the word surveillance criteria came to be. They said, okay, we're going to do a 2-tier approach, but it isn't for clinical purposes. It's only for surveillance criteria. Well, guess what? Every insurance company in the United States took it as clinical, and now in the United States, you have to get an ELISA assay first, followed by a Western blot in your diagnosis. And if you don't want to go that route, you have to make a choice. You either go become an IDSA guy and follow the strict guidelines of them, or you have the guts to be people like Dr. Bach on my left here and be a member of ILADS, where you're willing to treat the patient on a patient-by basis, and hell or high water, you're going to treat them till they get better. Thank God we got doctors like Dr. Bach. So I put this up because Paul Mead, I was at a meeting in, Paul Mead's the head of bacteriology at the CDC. He was at a meeting in Dearborn, at Mass General, and a friend of mine, Alan McDonald, a pathologist, was sitting next to me at the meeting, and he hit me. He says, "Hey, what's that slide from? Where did they get that Western blot?" Whoops, excuse me. That Western blot's an IGeneX Western blot on a CDC slide that Paul Mead presented. So they'll tell you that IGeneX doesn't do a great job, but they sure as heck like my Western blot. So this is Jennifer Halsey from your arthropod disease program over in Albany, saying that we're not even looking at the right proteins anymore. How can we expect to be getting the right answer? In Lyme disease, your bug has it's smart, it's big, it's 975 million base pairs big. Your human genome is about 4 times that size. This bug has 126 genes on it. It will outsmart your immune system. And over the course of the time of its being in your body, it changes its outer surface proteins, it goes into hiding, it comes back, all in response to how well your immune system reacts to it. So we have a bunch of different tests that we think, and we have found over the last 20 plus years, that do work at different phases. So early on in the disease, when the bug is high, you could do a PCR test if you could find it. PCR, the bug's hiding, you may not get it. But early on at the site of the bite, we can usually get a PCR positive. That's looking for the DNA of the bug. Later on, you can go on and do a T-cell response or an immunological serology test, such as a Western blot or our immunoblot, and those will be okay. So what we're doing in serology is looking for an antibody-antigen reaction. Your body produces antibodies against this bug. We throw those into the tube or into the test. We throw in the antigens that we think are specific for the disease. We look for an immune complex to form, and if it does, voila, we usually get a positive when we use a second antibody on it. Now, since my wonderful time in here since 1991 at UConn, I have seen studies that show all of these proteins associated with Lyme disease. Well, back in '94 at the Dearborn, Michigan meeting, and those of you who are overachievers, there's a 400-page article from the from the Library of Congress that explains all this data, and you can get it from them if you're a real overachiever. But here's the short thing, is that all of these proteins can show up in Lyme disease, but many of them are there because of its Epstein-Barr or some other co-infection issue that you've got going on. Here's a Western blot and why doctors like IGeneX, because we go into the gray zone. The zone where the patient may be sick, they're not producing a strong immune response, they need to know we're in the middle. Now, I point this out because if you look down at 39, which is a specific band, I keep doing that, specific band for Lyme disease that doesn't cross-react with anything else. If you look at the band, line 8, those are patients, lines 2 through 7, 1 and 8 are controls. Every Western blot's the same by CDC criteria or by to get clear licensing of it. So what happens is, take that 39 control and look across, going across the patients from 2 to 7. If you look at patient in number 2, well, that's stronger than the 39 band, so that's positive. Well, what do you do about 3 and 4? Something's going on. The body's trying to produce immune response. You're going to get a negative test here from Quest, LabCorp, or everybody else. And it's going to also be negative by IGeneX, but we are going to say IND, indeterminate, so your physician can make a decision. Okay? And we're trying to stay ahead of the plague. We now have recombinant proteins that seem to be significantly more specific than using antibodies. Now that we have the DNA capabilities to do that, we now have proteins that are absolutely specific for all of the sensu lato strains of Lyme. IGeneX has developed those with our friends over at Stanford. We are in Palo Alto, so they're right across town. And they got a couple of good molecular biologists over there. So we got some really good recombinant proteins that are specific to all of the sensu lato strains, i.e. the 7 major strains of Lyme. How good is this test? Well, when we compared it, or the CDC compared it rather, they found that it didn't cross-react. The sensitivity and specificity of this test, specificity approaches 100%, and sensitivity approaches about 90%. So we've got a new test, but we've got to try to stay ahead of the plague. You've got all of these strains up here in your state. The tests that you get from Quest LabCorp will not test for everything. None of us will test, and be advised, relapsing fever. It's not allowed to be tested for here in your state yet. You have a problem coming, and the Miyamoto strain, that second one down, is transferred from mom tick to about 1,500 of her eggs. So this is not something we're going to play with. I can tell you that in Maine, 25% of the the moose in northern Maine died last year because ticks infected with Miyamoto, 3,000 to 4,000 of them per moose, were killing the moose. It's now an epidemic in Connecticut, an epidemic in Massachusetts, and it's approaching there in other places. But of course, we can't test for it here in your state, so I'm not sure where you stand with this, other than knowing that the deer do cross over your state line because there's nobody to stop them from coming. So there are the different tests when you want to use them all. Western blot's still the best test here at the United States for hygienics. Remember that we test for all of the outer surface proteins that are specific. We don't pull out 31 and 34, which the CDC requires pulled out of every kit that's used. We are licensed fully in your state to do testing. So yes, the doctors who try to tell you they don't use hygienics because I can't use it, it's not licensed, that is a smokescreen. We are fully licensed. Most of our testing is available to you. And it does, and of course, doctors like Dr. Bach and others who do treat chronic Lyme use this routinely. Okay, so here is the co-infection map. You heard about it, the other things that are there. This is our map of the patients we're seeing with other co-infections besides Lyme, not just tick-borne relapsing fever, but also Bartonella, or cat scratch they call it, mycoplasmas, anaplasma, and of course Babesia, which is very big. So everyone likes to say they do testing as good as hygienics. You don't need an hygienics test. I'm going to tell you that I definitely do testing as good as hygienics, so you should probably consider us. Thank you for listening. So you mentioned earlier Richard Horowitz's book, MEMSID's questionnaire. I'm asking you to please go to his website, download the free MEMSID's questionnaire, take it in, and let all of your doctors see what the symptoms you've got. Because if you walk into a doctor just saying, I think I got Lyme disease, he is not going to have the time to go through all the symptoms that he should. If you have the form in front of you, it's a 5-page piece that actually shows all the symptoms. You have a much better chance of getting him to believe you actually have Lyme or one of the co-infections. So again, thank you for listening. Stay out of the woods.
Yeah, I feel like I've been here before. Just to ask, how many people have or have Lyme disease? Okay. And how many family members have Lyme disease? Okay. Lyme disease, as Holly said, she said most of the things on this slide. During my training, which was way back, syphilis was the great pretender. Now Lyme is the great pretender. It can masquerade as many diseases, many symptoms. And the EM rash and swollen knee myth is wrong. I've had an infectious disease who saw a patient of mine who had rheumatoid arthritis, and I diagnosed the cause as Lyme, and he brought a book out and showed her and said, look, no EM rash, no swollen knee, you can't possibly have Lyme disease. So what you can do is be an advocate and go to your doctor and make sure he or she listens. In medical school, we learned that 90% of the diagnosis is given by the patient's history. The lab is supportive. Lyme is a clinical diagnosis. Now, Holly or Bob mentioned about Dr. Masters in Missouri. He had a statement that I've used a lot called the absence of proof is not proof of absence. So absence of a test does not mean you don't have Lyme disease. Tick-borne disease is a clinical diagnosis. So I always tell patients, be in the question. Your doctor should be always questioning things. Unfortunately, doctors either like to know stuff or tell you what they don't know. They don't want to be in that area of that they don't know, and they're exploring what the question is. So I don't treat Lyme disease. I treat the person who has Lyme disease. So tick-borne disease, in case of chronic symptoms in this area, you have to think of Lyme disease or tick-borne disease. We're in an endemic area, and you need to review your diagnosis. If you have a diagnosis of chronic fatigue or fibromyalgia or chronic headaches or GI complaints, you have to think of Lyme. So you have to look for patterns of Lyme and co-infections, which I'll discuss. Lyme, Bartonella, Babesia, Mycoplasma. As Holly said, EM rash, less than 50% of people I see have it, so you can't hang your hat on that. Testing is insensitive, as Bob and Holly both said. So you have to think of the chronic disease, fibromyalgia, chronic fatigue syndrome. It's, you get the infection and it leads to immune problems and fatigue and fibromyalgia, along with many other things. So Lyme is a multi-system disease, and the reservoir is the nervous system. 2 weeks after you get Lyme disease, it's going into your brain. And there was a recent study in 2018 that showed it forms biofilms which make the antibiotics harder to get at it and make for persistence of the illness. Insomnia, fatigue, very big symptoms. Migratory joint pain. If you have joint pain, as I'm taking a history with someone in my office and they say I have knee pain, then I said, when did you have shoulder pain? Well, how'd you know? Because you have migratory pain. It can be in your knees for 3 days and then all of a sudden go somewhere else. Or it can stay in your knees. Headaches, usually Lyme is posterior neck and posterior head. Fevers, sweats, temperature dysregulation, common in Lyme and co-infections. Muscle weakness, muscle twitches. Cognitive dysfunction, very common in Lyme disease. Anxiety, depression, and I've seen a lot of cases of psychosis where someone has no history and all of a sudden they have psychotic symptoms where they're out of touch with reality. Numbness, tingling, dizziness, and palpitations and GI problems. Bartonella. Bartonella is another organism that goes into cells and has even more neurological symptoms than Lyme disease, and together they amplify. You could have central nervous system, that's the brain. You could have irritability, you could have tremors, you could have seizure, you could have encephalopathy, you can have cardiac symptoms. Bartonella particularly goes to the GI tract and to the lymph system. So if you start seeing this constellation of palpitations, nerve problems, tremors, seizures, been to this neurologist, been to another neurologist, been to a cardiologist, you have to start thinking, what's going on? Sorry about that. I should have shut it off, sorry. Okay, in Bartonella, you get skin rashes, and you also get things that look like stria, like serpiginous rashes that can be on your flank or your back. And you also get things like sole pain, shin pain, and of course, joint pain. This is a 32-year-old male. I figured I'd give you a couple of cases to illustrate things. Had vertigo attacks, headaches, fatigue for 5 years. He was treated symptomatically for vertigo, symptomatically for migraines. In his history, he took Cipro, that's a quinolone, for his prostatitis, and he felt great. When I tested him for Bartonella, he was positive both IgG and IgM, and he was treated with Bactrim DS for 12 weeks, and he was fine. Now, I just want to say a thing about, I used to use quinolones, but lately I'm a little leery of them because they can cause problems with energy and mitochondria and also tendinitis. But they are a good drug if you really need them, but you have to watch out. Babesia is a malaria-like parasite, which I'm seeing more and more in patients. It impairs the immune system. Now, a lot of times the infectious disease will say, well, did you do a blood smear? Blood smear is usually mainly positive in the first 1 to 2 weeks. I find the best test for that is a Babesia fish, which is done by Igenics. Treatment of Babesia can be 3 weeks or it can take 3 to 4 months. And the medications I've listed, that's Mepron, Tindamax, Plaquenil, Artemisia, and Bactrim and Coartem, and also herbal treatments like cryptolepsis and things like that. Babesia symptoms can be cough, air hunger, shortness of breath. And I see that a lot. Someone who's been worked up for pulmonary, cardiology, and they have this chronic cough, you know, and maybe it's allergies. But when you start to see it with fatigue, sweats, intermittent chills, usually the chills in Babesia are every 2 to 3 days, you may see that. Also associated with Babesia is anxiety, panic, chest pain, palpitations. I see a lot of POTS, which is postural orthostatic tachycardia syndrome. Headache. The headache in Babesia is on the top. The one in Bartonella is mainly frontal, and the one with Lyme. But that's just guidelines. They could be anyway. And in Babesia, you see a lot of mental, emotional, and cognitive symptoms. You also can see GI symptoms, which most people feel could be Lyme or Bartonella. But I had a case out in California that I worked with this GI fella, and a 30-some-odd-year-old actress had horrible SIBO, horrible GI problems. She turned out to have Babesia. When the Babesia treated, she was fine. Now this, again, a 58-year-old woman was in good health, had a tick bite in June of last year, then had flu symptoms, no EM rash. She then had the onset of fatigue, migratory arthralgias, neck stiffness, fatigue, headache, night sweats, poor balance. She saw her primary care who referred her to infectious disease. After the referral, she then had ringing in her ears, loss of strength, and word retrieval problems. Her tick-borne testing was negative. The ID made no diagnosis and referred her to rheumatology and neurology. I consider that the usual suspects. She asked for antibiotic treatment, and her infectious disease specialist stated, "No, you're not dying. Why would I do that?" Her ID in her chart said, "I reviewed the lab serology with the patient who verbalized understanding. I cannot find any infectious, environmental, rheumatologic, viral, parasitic, or other cause for this patient's current experience." And you see this, they kind of cover their ass, so to speak. 1 month after treatment, she was 90% better. This 15-year-old girl had psychiatric symptoms for a year and a quarter before I saw her. She had a history of anxiety, depression, personality disorder. She was on 4 different psychotropic meds, including trazodone for sleep. She was positive for Bartonella Ehrlichia, but negative for Lyme. But when I treated her for Lyme with IV ceftriaxone for 10 weeks, she had a complete recovery. Interestingly, her parents came in the first visit. I asked them, "What do you know about Lyme?" They said, "What? Nothing." By the end of the 3rd or 4th visit, they were so up to speed on Lyme. Okay, I'm going to skip this. Okay. Other aspects of tick-borne disease that you have to know about, it was mentioned before, co-infections, biofilms, Herxheimer. When you treat someone, they may get worse, and you have to coach a person and say, "Look, you may get worse in 2 or 3 weeks or in 3 days." You have to think of your immune system. You have to stay away from sugar. You have to watch stress, and you have to watch sleep. You always have to look at your endocrine system, because that's your organ of stress, which produces cortisone, and your thyroid, and of course, nervous system, like I mentioned. I'll put this up. I'm not going to go over it. It's just the criteria for IV, because I treat people with oral medicines, IV, herbal, and then I'm going to go over some of the more recent treatments that I've been using. Okay, I just wanted to put this chart up because this shows the difference between ILADS and IDSA. If you have a tick bite, I find that I've had people, and it's in the literature, that after 6 hours of a tick being on, you could have Lyme. IDSA goes by 36 to 48 hours. For a diagnosis of the EM rash, ILAD says at least 6 weeks plus. IDSA says 3 to 4 weeks. For early Lyme, which means you could be diagnosed clinically, again, I would treat for at least 6 weeks. IDSA says there's no positive test, you don't have Lyme, we're not going to treat. Persistent Lyme, ILAD says treatment depends on the case. IDSE says post-treatment Lyme disease antibiotics are not justified. Co-infections, ILAD says if you have a pattern of co-infection, even if your test is negative, we will treat. IDSA needs a positive test to treat. So conventional treatment, antibiotics, different forms, intracellular forms, cyst forms, use different antibiotics. We treat for 6+ after symptom resolution. So if I treat someone and they're better on the antibiotics 3 weeks, I don't stop because that person's could relapse. And also if they have a herx, 3 weeks into treatment, I don't treat for 6 weeks. I'm gonna probably treat for 8 or more weeks. And then I also, instead of daily treatment, I may step down and go to pulse treatment where you have treatment with antibiotics or herbals Monday, Wednesday, and Friday. So integrative treatments, you always have to assess what your function is, both in terms of infection, your metabolism, toxicity, the status of your immune system, inflammation, the status of your nervous system, cognitive function, energy function, and assess the past treatments and your response. So it's not a cookbook. You don't give someone an antibiotic and say, I'll see you in 6 months, goodbye. And integrative treatments, diet, very important. You also have to do an elimination diet because you could have food allergies. Low sugar, like I said. Yeast-free because you're treating with antibiotics. In some cases, you got to think about yeast. Herbal treatments, homeopathic. Detoxification, always important. Herbal regimes. Treatment of neurotoxins and mold is a whole other thing. And lately, the treatments that I've been using is ultraviolet light, which kills pathogens, helps the immune system, and ozone, which kills pathogens. Okay.
Hi, this is Kathy Wallace, founder of Upstate New York Lyme Disease Association, and I have the questions behind the scenes, so I will be asking. I do want to thank Wendy for bringing this together today. She's been very instrumental in helping with Lyme. So this first question would go to Dr. Stephen Bach. How do you get the Western blot test when your Lyme test comes back negative and your doctor tells you it's not needed?
Well, they call that a reflex, and when I order it, I always order a Western blot, so I don't ever order where they just do ELISA, so my lab knows that I'm going to order ELISA and a Western blot.
This next question would be for Bob. Should we travel to another state for testing for treatment of Lyme disease, and if so, which is the best state to go to?
Oh, loaded question. No. Actually, our Western blot is probably the best test in the United States right now for testing and treating testing for Lyme disease. The treatment protocols are pretty much opened up now because of ILAD's interpretation and their help. Again, our Western blot is a lot different than the one you get from Quest or LabCorp. We're not going to pick up most of the patients of Lyme if you're testing with your local lab. And I didn't bore you with the Mardex blot, which I developed, which is what Quest uses. So I won't bore you with the science behind it. But I can tell you that the Western blot from IGeneX is going to give you the right answer. It is licensed in your state. Okay, so you can use it. And most of the tests, like our fish testing, as the doctor mentioned, are licensed here. And you've got some very, very, very competent ILADS physicians like the one on my left in this state. You really don't need to spend a significant amount of time just trying to find another place to treat.
Thank you. Do any of you have any experience with Lyme disease being transmitted by any other insects, such as a mosquito or a flea?
So I'm going to answer that from the perspective of extensive reviews of the scientific literature. So there have been reports in the scientific literature that mosquitoes might harbor the microbes and perhaps other blood-sucking insects might have the microbes, but there's no evidence that they can transmit them to humans. That's a really important thing because it's part of a natural life cycle that involves the tick, the microbes in humans and other animals, and it's a complicated cycle. So just because they found the bacteria that cause Lyme disease in mosquitoes in Europe does not mean that they are able to transmit the bacteria and it can establish itself as an infection. One of the reasons why ticks are so successful at this is because they inject us with this chemical, well actually this cocktail of chemicals that inhibits our immune system, that causes us to not feel their tick bites, that sets up the stage for the infection to disseminate. That's a relationship between the tick and the microbe that is probably not the same relationship that you would find in other microbes. So there is, I'm confident to say that as far as transmission, tick-borne transmission or insect-borne transmission, that ticks are it. The thing that Mary Beth's book will tell you is that ticks are everywhere. They are expanding in numbers and in locations. And so we are at a significant risk of ticks, whether you see them or not. They are masters of stealth and find places to feed on you where you might not even know that you have been fed on by a tick. So they're remarkable enough without looking for other kinds of vectors. And I actually just want to say something in response to, well, you're going to say, right, that you can send ticks off to be tested for microbes at IGeneX Lab, but I also want to say from the perspective of both of the two questions, New York State physicians can be educated. They are following medical guidelines because that's what they do. And you as a patient or as a parent of a patient have every right, and by right I mean you have rights, to say to your physician, I would like to discuss this, I would like to be participating in my treatment decisions, and I want my blood test done through IGeneX, which is something that New York State physicians do not understand, that IGeneX Lab is licensed by CLIA, which is a accrediting organization nationally, and licensed specifically in New York State. So a doctor cannot tell you they can't order an IGeneX test because they absolutely can. They may need to be educated on that. So you need to empower yourself. Knowledge is power. So learn this information so that you can take that information to your physician that you already have a relationship with, so that that you don't necessarily need an ILADS trained physician if you are aware of what you need to do.
Also, yeah, I'm head of the medical education of ILADS, and one of our tasks is to train physicians. So you can go to your physician and say, ILADS is a place that you can go and take a course for 3 days and get your education very quickly and at least start the program going. One weekend won't do it. Treating Lyme disease has a lot of nuances, and you have to really be aware of that. But if you get your doctor to go, I've seen doctors who come there who are completely negative on Lyme disease, and as soon as they finish the weekend course and say, like, I can't believe it, the course blew me out. So you can always tell your doctor, I want you to do that.
I would just add that there are two dueling sets of guidelines for Lyme disease: the Infectious Diseases Society of America and the International Lyme and Associated Diseases Society, ILADS, the ones that Dr. Bach follows. And you can tell your doctor that the IDSIA guidelines, the Infectious Diseases Society of America, which basically rule Lyme disease care worldwide, haven't been updated since 2006, since 2006. They are old, they are outdated, they are based on old science. The ILADS guidelines were published in 2014, much newer. They've met evidence-based standards. So you can tell your doctor, here, these are newer guidelines, they're more up to date, they have been endorsed. The other ones, out of date.
I had a case a couple of years ago where somebody came to me and the testing was pending, but she was so bad that I said, look, I think you have clinical babesia. I'm going to treat you with this. You may get a herx, call me, you know. She had horrible anxiety, chest pains, palpitations. She went to the hospital. I got a call from infectious disease saying to me, how dare you put this person on Mepron? Why did you do that? And what I did is just, oh, I hear you. I knew she'd be back. You know, she got out of the hospital, she came back. I continued the treatment with Mepron. She totally recovered. But her testing came back negative, and she responded to clinical treatment of babesia. So,
All right, this next question would be for Dr. Bach again. Is there a connection between Lyme disease and autism?
well, yeah, I think, you know, in terms of, so autism is really increasing. So, and there's so many things that overlap, encephalopathy and things like that. So I think if a child has autism, with the many things that can cause autism is environmental exposures to things, methylation problems, but then if they have a Lyme infection, that can really make the autism worse like that. But the last time I had spoken to Dr. Jones, who's one of the experts on pediatric Lyme disease, he said very few of his cases had autism from Lyme disease, congenital Lyme disease.
This next question, I personally do like this. If this is so critical, why aren't drug companies trying to develop a drug to profit from Lyme disease?
Because we don't know what Lyme disease is, because we don't have an accurate diagnostic test. And what we have, Lyme disease falls under the realm of infectious disease medicine, just like other bacterial infections do. What infectious disease docs have at their arsenal are antibiotics. Antibiotics do not effectively treat this disease for most patients. And unfortunately, we got nothing else. And until we convince those docs that this is an infection by bacteria and that they are resisting the effects of antibiotics, and this is different than antibiotic resistance, this is tolerance, meaning you're going to treat me with antibiotics? I don't care. Until we come up with, we get past that, we're not going to be able to do research, meaningful research, to come up with ways to effectively treat this disease. So in the meantime, you need to treat the disease until you are better. And the one-size-fits-all 3 weeks of oral antibiotic is not an effective treatment for the majority of patients. But that doesn't mean you can't say to your doc, look, there's other guidelines. I really would like you to read these. I would like you to consider a round of IV Rocephin, or this or that. And it is within your purview as a patient to say, I have the right to informed consent. And what that means is they're supposed to inform you, and you guys discuss what the best treatment options are, and then decide collectively. So those two words, informed consent, are very powerful. Do not be afraid to use them.
Now, about 8 or 10 years ago, someone lectured at one of the ILADS conferences, and he was saying, like, there are bacteria that have been around for 4 billion years. There's bacteria that eat rock. There's bacteria that eat oil. He says, so you can imagine the spirochete is on a train, you know, and you're hitting that train with antibiotics for even 6 weeks, more than the 3 weeks. And the bacteria is saying, I'll just get off at the station. I'll wait a couple of months. I'll get back on. Now, I, through the years, have really changed my thinking. Like, to me, Lyme is, everybody knows herpes. Herpes is a virus that goes into your nervous system, and then if you get stressed, or if you go in the sun or something, you know, you can have a herpes outbreak. Well, Lyme is a bacterial, is like a herpes. In other words, the immune system walls it off, however you do it, if you beat it down with antibiotics or herbals or IV or the ultraviolet light. And then you have to watch yourself. You have to watch your immune system. You have to stay away from sugar. You have to not stress yourself. You have to sleep. A case in point, I told a couple of nurses that I treated, your Lyme is much better. Don't get a flu shot. So then I got a call about 6 months later, you know, different times they called me and they said, you know, my symptoms are back for 2, 3, what's happening? And I said, wait a minute, and it got better. And then as I was talking to them, I said, what did you do? And they said, nothing, I was just on the floor and, oh, they were giving out flu shot. Oh, you told me not to get a flu shot. It's almost like you get the flu shot, your immune system goes to take care of the prevention from the flu and leaves the door open, and you get the Lyme that kind of comes back. But a lot of times, if you just watch yourself and go back to what you're doing, it'll calm down and you may not need antibiotics. But what I'm trying to show with that is that there's a balance between the infection and the immune system and the hormone system and your body. And Lyme, in some people, really makes them look at their body and really change their thinking. It's
Real quick, this is the perhaps cynical view, but the pharmaceutical industry is probably very happy with the status quo right now. They would much rather treat a chronic illness. They just don't call it chronic Lyme disease. They call it fibromyalgia. They call it chronic fatigue syndrome. They call it even multiple sclerosis. There are a lot of different diagnoses that are attached to people who actually have Lyme disease. It should be ruled out. But the long and short of it is, there's not much incentive on the part of the pharmaceutical industry to deal with this.
Next question would be on bee venom therapy. Is that a cost-effective therapy in healing Lyme? Does it work?
Well, bee venom therapy definitely can affect the immune system, and it's useful in modulating some of the symptoms of Lyme, like especially neurological, like multiple sclerosis, headaches, and things like that. To me, it's one of the things you could do. It's not a standalone treatment for Lyme disease.
Why are these tests not allowed in New York? I would assume that's for Bob.
What happens in your state is that you have a Department of Public Health that doesn't believe any other state's departments of public health. So what happens is they have to run the test on their own, in their laboratory, their way, and correlate it in a double blind against the test and the technique that you've got with the different new test. And New York Department of Public Health doesn't have the money to continually build all these new and chase all these tests. So since they can't, they say you can't have the test until we have time and energy and the money to actually validate the technology and the test. And that's why.
Also, years ago, I looked at that problem, and what happened is New York State wanted to fund Wadsworth, which is Albany lab, the Department of Health's lab, and the federal government said, well, you can't do that because we regulate labs with CLIA. And the New York State Department of Health said, well, what can we do? And the federal government said, well, if you had a different criteria, you could get it out of our system. So New York system was then allowed to say, okay, we'll decide on the efficacy of that test. So what Bob says is how they go about doing it, but really, if I go to a conference and I walk at the tables and I see like this new test or this new treatment, you know, I can't do it because it's not allowed in New York State. So it really paralyzes a lot of physicians. I have ways that I get around it, but it's just a horrible problem.
And realize that IGeneX is CLEP, that's a Clinical Laboratory Evaluation Program, New York State CLEP. They are certified, they are licensed through our verification program. So that means New York State stands behind the tests that IGeneX does in terms of their reliability and accuracy. And that is what I'm saying. Our New York State physicians don't understand that or don't recognize that, or for whatever reason, but that is something, use the words CLEP, Clinical Laboratory Evaluation Program, New York State. Department of Health says this laboratory is reliable, gives reliable and accurate test results. Your doctors can order them, and your doctors can use the results for insurance, to treat you, all that should be reimbursed under insurance. Again, this is empowerment on your part.
Thank you, Holly. And if you really want to, just go to our website and download the New York State license. It's right there. You can walk into your doctor with it and say, here, this lab is licensed. What do you mean you can't offer it? Okay, just download it and you give them to that and see what they say.
The next question, can Lyme disease cause plaque on the brain?
Oh, yeah, Lyme disease can cause cognitive dysfunction, dementia, and long-standing Lyme. It's the same pattern as syphilis, is. And what happens, like I mentioned, in 2018, there was this article saying that after the infection is in there, biofilms are forming to actually hide that bacteria while it causes inflammatory changes, which are what plaques are.
The next question, what is a EM rash?
That's the erythema migrans. That's what Holly was talking about, when you get the bite and it forms at the site of the bite. But it also can form at other parts too. You can have someone who has multiple EM rashes, even though they didn't get multiple bites.
So it's a perfect bullseye. And what the science says is the perfect bullseye right around the tick bite occurs less than 10% of the cases of Lyme disease. Less than 50% of everybody develops any kind of rash. So the most common type of rash, skin rash, is a solid red raised rash that looks like a spider bite. And so most physicians are waiting to see the bullseye. They're focusing on the bullseye, and they will overlook a solid red raised rash. My daughter's situation, there was no rash. And that was because she was bitten by an adult stage tick, where the association between a bullseye and that stage tick has never been made in the research literature, ever. So when I'm talking about statistics, if you have a perfect bullseye, a perfect target rash, you are at the highest likelihood of getting an accurate diagnosis and early treatment. That's 10% of the cases. If you have a solid raised rash or no rash, you are very likely to not get diagnosed early. The bacteria will spread, and at that point, it's a whole different clinical scenario.
At this time, I just want to put a quick commercial in for our local support group. We do meet at Wendy's store every third Monday of the month at 6:30. Everyone is welcome, whether you are ill, you have a family member, or you would like to learn more. I want to thank the four of you for being here, and I will leave the floor to you if there's anything else that you would like to add to the audience. So I will let you end the show. Thank you.
I'll just throw this out. We need to mobilize. We need to make noise. We need to write letters to the editor, to call our legislators, to demand the $1 million that was dropped from the New York State budget get restored. We have to let the powers that be know that this is not acceptable, that we have thousands upon thousands of cases in New York State every year, many people remaining ill, and they go to the doctor and they can't get diagnosed, or if they're lucky and get diagnosed and stay sick, they can't get follow-up treatment. Doctors don't know what to do about it. They haven't been told what to do because they're following those prevailing guidelines. Something needs to change on Lyme disease, and a public outcry will help.
That lady's saying how much Ken Bach saved her husband's life.
Yep, shout out to
And
Dr.
Steve
Bach.
here. The boys.
So my last word is, all those mamas out there, let your inner bear shine.
Hi. I sent a question by text, but I don't think it went through. Is there any utility in the C6 peptide test? somebody had mentioned that to me.
Okay, in 1996, I helped develop that with Andy Levin at the Massachusetts General Hospital. We developed it thinking it was going to be better than the standard ELISAs, which were available through the 1990s from places like Meridian and other labs. The C6 ELISA peptide is okay in early Lyme disease. We knew it right up front. And until about 2003, we were hoping it was going to be the panacea. It never proved to be. Andy went off and formed his own lab after he left the MGH, his own company called Immunogenics, and they bake the kit. They sell it to a bunch of people, including LabCorp. We have it available at IGenix. Again, in the first 8 to 12 weeks, it's a pretty great test. After that, I worry about C6 showing up in late disease. It's hiding or it's crossed over by quorum sensing. So no, not in late Lyme, early Lyme. And you can always go, even though many of us hate Alan Steer, I don't. I think he's a pretty good scientist, even though he's done some nasty things to the rest of Lyme. His science on the C6 ELISA published in Infection back in 2005, pretty much put a nail in the last coffin for it. It's about 20% better than using a standard ELISA. But ELISA is less than flipping a coin, so you're not getting any great tests there. Okay?
You know, I agree with that, except for the fact that I've done C6 peptides, and it'll be, if it's negative in someone, it's useless. If it's positive in someone, you can actually use it quantitatively, because if it goes down, and that could take a year and a half, you could tell me if I'm right on that. And then 4 years later, if they get symptoms and I do a C6 peptide, in them, it'll come back. So that's the usefulness that I find with it.
It's just it's not going to pick it up all the time.
Yeah.
Okay?
Can I ask a question? Okay. So does anybody have a recommendation about when there's Lyme disease and mold as a confounding factor? And either have done work with that or recommendations?
That's about a 2-hour topic.
Thanks.
Thank you.
Well, they all deserve a standing ovation for what they've done. They've done a great job, and thank you, folks. My question is, can you obtain Lyme disease from other than the tick, like the cockroaches, other insects, mosquitoes, and so forth? Pardon me? Oh, I'm sorry. I didn't know that you had, I got here a little late.
The science says no.
But would it be communicable between person to person?
The science says maybe.
Maybe. Oh,
Maybe.
my. Okay, thank you so much.
Yeah.
Very good job.
Okay.
Thank you.